Good morning, guys. Welcome back to acid-base balance. We'll start with the second class of acid-base balance today. Now, in the previous class, we we saw the Bronsted-Lowry concept and the acid-base conjugate concept. As the weak base, it would give a strong acid, and a weak acid would give a strong base, and the vice versa is also true. So we saw the Bronsted-Lowry concept. Then we saw how pH as a concept arose, and how Sorensen, the scientist, worked behind it to develop it into a pH, the concept of pH. And we saw the buffers, the concept of buffers, defining buffers and all. And we also saw usually a combination of a weak acid and a strong base is found as a buffer. Acetate buffer is an example. And also we saw the three defenses employed by the body to fight against the acid-base imbalance in general. And the three defenses were the blood buffers, respiratory regulation, and renal regulation. We covered the blood buffers in the first line of defense, which is the blood buffers, in the last class. So today in this class we'll be dealing with the respiratory regulation and the renal regulation. Okay. So as I told in the last class, the three line of defenses are these. And the first line of defense is the blood buffers is employed as an emergency response. Respiratory regulation comes into action after 5 minutes, and the renal regulation kicks in after some days, and it is supposed to be a permanent solution to whatever acid-base imbalance has occurred in your body. Now, to go on to the second line of defense, to start with that, the respiratory regulation have two main mechanisms. It usually acts first with the help of hemoglobins, and also it has a process known as regulating the respiratory rate. It can actually increase the rate of respiration or decrease the rate of respiration. So first we'll see how does it act with the help of hemoglobins. We already saw that hemoglobins are actually are proteins which are actually having a buffering capacity by itself. Here we'll see how it helps actually in association with respiration. How does it actually help us to fight off the acid-base imbalance? Now, coming to the first thing, with the help of hemoglobins, it works effectively with the bicarbonate buffer system, and it eliminates carbon dioxide by the reversal of chloride shift. You might be familiar with the concept of hamburger shift, chloride shift, and all from your lower classes when you learn about respiration. Now, this is the same thing mentioned here. Here, what happens is that hemoglobin can eliminate carbon dioxide from our body by a process known as reversal of chloride shift. Now, you should understand that when I say eliminating carbon dioxide, now usually carbon dioxide in our body or in the body fluid is actually seen as carbonic acid. So when you remove carbon dioxide away from our body, that means you are getting rid of the acid from our body. Okay, so if carbon dioxide is existing as carbonic acid in our body fluids, when you expire carbon dioxide outside, you are actually getting rid of the acidic thing in our body, which is carbonic acid, right? So this is done by the reversal of chloride shift, which we'll see just now. Now, this diagram on the left side is actually showing the activity at the cellular level. Okay. Now you can see here at the cellular, after the cellular metabolism, the carbon dioxide is produced at the cell level, and this carbon dioxide produced in the cells and tissues will actually diffuse through the capillary endothelium, and this carbon dioxide will enter the RBC. The RBC, inside the RBC, you can see the carbon dioxide and the water combining to form carbonic acid in presence of carbonic anhydrase. This carbonic acid actually will split into bicarbonate and H+. Now, this is what happens inside the RBC. Now, at the tissue level, you can observe that at this area, you can observe that the bicarbonate will go out, will diffuse out of the RBC, and in order to maintain the electrical neutrality, the chloride will diffuse back. Okay, so once the bicarbonate goes out, the chloride will diffuse back. Now this bicarbonate is present in the plasma now. It is no more part of RBC. So bicarbonate is now there in the plasma, right? Now this and the H+ which is there will actually reduce the hemoglobin, and the reduced hemoglobin is stored inside the RBC. Now this happens at the tissue level. Now we know that the RBCs take carbon dioxide from the tissues and come to the lungs and release the carbon dioxide and absorb oxygen and come back. Now let's see what happens at the respiratory level. Okay, At the lung level, what happens? This RBC, when it reaches the lungs or the alveolus, what happens? This the reverse of this reaction happens here. Bicarbonate comes inside, diffuses inside the RBC. Chloride comes outside again to maintain the electrical neutrality. The bicarbonate will combine with the H plus, which is donated by the oxidized hemoglobin, which actually combines with oxygen and releases the H plus. This release H plus and HCO3 minus will combine to form carbonic acid. Again, the same enzyme acts to split it also, which is into carbon dioxide and water. This carbon dioxide will diffuse into the alveolus and finally into air outside. So this is how we expire carbon dioxide, how carbon dioxide is thrown from the metabolic reactions through the RBC, and it throws out the carbon dioxide like this. So when you do throw out carbon dioxide like this, I told you we are actually getting rid of this acid, this carbonic acid which is there inside the RBC. So this is an excellent mechanism to bring down the acid level. Whenever the pH goes down or the acidity in our body increases, the respiratory system will try to get rid or bring back the pH, bring up the pH back to the normal by actually getting rid of these acids by this mechanism. It will try to throw out the carbon dioxide. So this is one mechanism with the help of hemoglobin, and this transport of carbon dioxide in the form of bicarbonate from the tissues to the lungs is known as isohydric transport. Just understand that term, such a term is there. Isohydric transport is the transport of carbon dioxide in the form of bicarbonate. Now, to sum up the reactions which happen in the tissues, these are the reactions as I told you. Due to metabolic reactions, carbon dioxide and water produced are combining to form carbonic acid. Carbonic acid will again dissociate into bicarbonate and H plus. H plus will combine to, with hemoglobin to form a reduced hemoglobin. And in the lungs, we have this reduced hemoglobin combined with oxygen to give oxygenated hemoglobin, and H plus is released. This H plus will combine with HCO3 minus giving H2CO3. H2CO3 will dissociate into H2 and CO2, and the CO2 will be thrown out of the lungs during expiration. So that is all regarding the first mechanism under the second line of defense. And the second mechanism was the regulation of the respiratory rate. As I told you, the respiratory rate is around 20 to 25 breathings per minute, or inspiration expirations per minute. So when you say regulation, that means either the respiratory rate can increase or decrease. So actually, as I told you, if you can actually adjust your rate of breathing, that can actually, if you increase your breathing rate, that is, if you breathe fast, you will actually pump out more oxygen, more carbon dioxide from outside your body, right? So if you breathe fast, more and more carbon dioxide are flushed out of your body. That will bring down your acid levels. On the contrary, if you breathe slowly, that means the carbon dioxide in our body is retained for a longer period. So that means your acid is building up in your body. So if you apply that thing here, that is what is meant by regulation of respiratory rate. Now, to understand this, you should understand that there are two chemoreceptors in our body. Chemoreceptors, from anatomy, you must be understanding that from the name itself, it indicates chemoreceptors. That is, these are receptors which can sense any change in the chemical nature of the body fluid. By chemical nature, I mean the pH of the body fluid. So if the receptors found in the peripheral and the central areas, which are known as central chemoreceptors and peripheral chemoreceptors. Central chemoreceptors are usually found in the medulla of the brain. Peripheral chemoreceptors are located around the carotid aortic arches and all. So if you have these two kinds of chemoreceptors in your body, whenever there is a pH change in these body fluids, these chemoreceptors can sense that change in pH. And these chemoreceptors will tell the brain or the lungs, the brain, to actually control the respiratory rate, okay, to increase or decrease according to the pH change. How does that happen? We will see now. Now, imagine at first condition, scenario 1 or condition 1, imagine if the acids are building up in our body, that is the pH is falling, right? Acids rise means pH will fall. So the chemoreceptors, which I mentioned just now, will sense this change in pH and actually ask the body to, or the lungs to, breathe fast. The brain will send a signal to, the chemoreceptors will send a signal to the brain that the pH is high, the pH is low because of acids building up. And the brain will get that signal from the chemoreceptor and ask the and the brain will in turn send a signal to the lungs saying that the respiratory rate should be brought up. So if you increase the respiratory rate, that is known as hyperventilation. If you ventilate very fast, it is known as hyperventilation, and hyperventilation occurs to pump out the carbon dioxide. So this is the response when an acids are building up in our body. So you can imagine when alkali rise, when the basis or the basic nature of the body fluid increases or the bases accumulate, what happens? The opposite will happen. When the pH rises, and that will tell the chemoreceptors, the brain via the chemoreceptors to decrease the respiration. So hypoventilation happens, and more carbon dioxide will be retained in our body. So these are the two mechanisms which are employed by. These are the main two mechanisms. So the two mechanisms we just saw, one was with the help of hemoglobins, with the help of reversal of chloride shift, and the other one was the regulation of the respiratory rate with the help of chemoreceptors. Okay, so yes. So the problem with the respiratory system is, yes, it responds immediately, maybe somewhat later after this blood buffers action, but the second line of defense acts immediately. But the problem with it is it cannot proceed to completion. It is not a permanent solution. So we have to depend on the third line of defense, which is more permanent and more effective when it comes to correction of the acid-base imbalance. That system of response is known as the renal regulation, which is a third line of defense. Now coming to the renal regulation, you must understand that, as I told you, it is a permanent mechanism. Now the attack strategy, see, the body is trying to ward off or fight against these acid-base imbalances. So the body or the renal system, which is a permanent system, as I told you, must have an attack strategy. The attack strategy are actually confined to these three areas. Okay, so the whole mechanisms employed by the renal regulation are actually confined to these three areas. Now, yes. So just make sure that you remember the reactions. So there are four main mechanisms which happens inside the renal regulation. So I'll be discussing each mechanism under these headings or these areas. Okay, blood plasma, I meant the blood plasma, which is the extracellular fluid of the blood, and in the tubular cell you have the tubular cell and tubular lumen are part of the kidney. As you might remember, the nephron has a distal tubular, the proximal convoluted tubule, distal convoluted tubule, and the collecting duct. In those ducts and the tubules, you have the lining of a tubular cell, and the lumen where the urine formed is known as tubular lumen. So these two are part of the kidney. This is the blood. So on that note, we can start discussing the strategies. So in the order of importance, the first strategy, and there are four strategies. The first strategy employed is the recovery of the filtered bicarbonate. Second one is the excretion of titratable acids. Third one is the excretion of ammonium ions. And the fourth one is the excretion of free H+. Okay. Now, the first mechanism that we are going to discuss is the recovery of the filtered bicarbonate. Now, if you see this recovery of filtered bicarbonate, you will understand that this happens, as I already mentioned, the attack always happens in these three areas. So the first reaction that we are going to discuss is the recovery of the strategy is the recovery of the filtered bicarbonate. Now, as you know, as you might have studied in renal physiology, all the things which are filtered filtered by the nephron will actually appear in the tubular lumen. So the first reaction is the filtration of the sodium bicarbonate. So on a daily basis, after every reaction, after every filtration through the glomerulus, we have some amount of sodium bicarbonate filtered in the tubular lumen. This filtered tubular sodium bicarbonate is actually dissociating into sodium and bicarbonate. Now, as you can see, yes, sodium and bicarbonate is formed. Now, to start with, another reaction happens parallelly here also. Carbon dioxide and water will combine to form carbonic acid in presence of carbonic anhydrase. This carbonic acid will dissociate itself into bicarbonate and H+. Now, if you look closely, you can understand that the H+ which is formed here is actually crossing over to from the tubular cell into the tubular lumen. At the same time, the sodium is there and the lumen is crossing into the tubular cell. This is due to sym-porter which is there. Sym-porter means a transport protein which helps molecules to move in different directions, okay, the opposite direction. So once H+ crosses this side, at the same moment sodium can cross the other side. So this H plus which is there from this reaction and this Na plus which has been dissociated from this will combine again, this H plus and the HCO3 minus in this reaction will combine to form the H2CO3 here, and this H2CO3 again will be split into these two, and these two will diffuse into the back into the tubular cell. So in this reaction, the thing that is happening is this bicarbonate formed here will be again be transported to this bicarbonate into the plasma, and the sodium is also transported there, and this together will remain in the plasma, or it is collected or saved in the plasma as the sodium bicarbonate. So this is how the alkali is recovered in the plasma. Now let me tell you one thing. This is the first strategy that we are discussing. In all the four strategies, you can see the alkali being recovered in this side. Okay, it's easy to remember this. Now why I'm mentioning this again and again is that it is a very frequently asked question from this acid-base imbalance chapter is the renal regulation of body pH, and in that you have to write the four strategies along with the diagram. So if you understand, if you listen carefully, you can easily reproduce what is drawn here. You should understand the three attack areas: plasma, tubulus, and tubular lumen. All the reactions, most of the reactions are similar in all the systems. Once you draw and study maybe 1 or 2 times, you will be thorough with that. Okay, no issues with that. So you have the reactions going on like this. So this strategy is named as the recovery of the filtered bicarbonate. So we are able to recover this bicarbonate is filtered here first. Yes, the strategy started. We started discussing with the appearance of filtered bicarbonate, filtration of bicarbonate here, sodium bicarbonate. That filtered bicarbonate is actually recovered at the end, right? So that is why the strategy is known as the recovery of filtered bicarbonate. Now, one issue with this strategy is that it is not able to get rid of any H plus ion or proton from our body. Okay, by this mechanism, the only thing that is happening is the recovery of the filtered bicarbonate. No other excretion of H plus or a bicarbonate happens in our body because of this strategy. So this strategy is not so useful to get rid of H plus, but it is a very important mechanism which happens on a daily basis just to recover the filtered bicarbonate. Because I told you, one of the most important extracellular buffers in our body is the bicarbonate buffer. What makes my bicarbonate buffer so special is the abundance of alkali in the plasma, and this abundance is achieved by recovery of this thing using this strategy. Okay, and this is about the first strategy. And moving on to the second strategy, about the first strategy, some things that to sum up is that there is no net change in H plus ions, as I told you. There is no net gain of bicarbonate also. And it cannot correct acidosis, but it can maintain the status quo. Okay, if any diseases or acid-base imbalance occurs, this strategy is not going to help you. Okay, this strategy is just to recover the filtered bicarbonate, only that. Yes. Now moving over to the second strategy, which is the excretion of titratable acids. Now speaking about that, I told you again the reactions occurs in these three areas. The first reaction we are going to see here is water and carbon dioxide combining to form the carbonic acid. Same reaction occurs in all strategies. Splits into two. The H plus will cross this side, the Na plus will go that side. Now here the filtered component shown is disodium hydrogen phosphate. And disodium hydrogen phosphate, as you can see, the pH given is 7.4. This thing is actually dissociating into Na plus and NaHPO4 minus. Na plus will cross that side and H plus will come this side due to the symporter function. And this H plus will combine with the NaHPO4 ion which is formed in the form of NaH2PO4. You can observe the change in pH here, right? Sorry. You can see the yes. Yes, you can see that the pH 7.4 has become pH 5.4. That means it was filtered as a basic thing or not so acidic thing. It's a basic thing, right? Above 7. Now at the time of excretion, when it combined with the H plus thing, the H plus and this will combine to form this NaH2PO4, which is a very highly acidic substance, and the pH is found to be 5.4. So by this mechanism, we are actually able to get rid of a lot of acid from our body because we are excreting this substance from the urine, through the urine, which is around a pH of 5.4. So that will bring down the level of acids in our body, or try to increase the pH back to the normal by using this mechanism. So again, as you can observe, as in the first strategy, again we are able to recover the alkali on the side in the plasma. So these are the two advantages with this. We are able to excrete H plus and also recover the plasma, recover the alkali in the plasma, right? So yes. Now coming to, yes, so to sum up the second strategy, we saw that when the extra H plus is there in the body, we can utilize this. The body will utilize this strategy. Now titratable acid means it is a measure of net acid excretion by the kidney. Here the titratable acid is the sodium dihydrogen phosphate, which was excreted in the urine. Now here what happens? Net loss of hydrogen is happening. Net loss of H plus is there, and also we have gain of HCO3 minus also present. So you have net H plus, loss of H plus, and also net gain of bicarbonate. Yes, both these we are able to achieve. So this is the second strategy, right? Now moving on to the third strategy, which is the excretion of ammonium ions. We see that the whole reactions as what as I suggested in the earlier two strategies, it happens in the three areas, which is the plasma, tubular cell, and tubular lumen. As you can see, all the reactions which were originally seen in the other two strategies are also there. Now, what happens extra in this is the reaction. This reaction is what makes this strategy special. This reaction occurs in the tubular cell. Glutamine, which you will learn in the amino acid metabolism, glutamine is acted upon by this enzyme glutaminase, and it will split it into glutamic acid and ammonia. This released ammonia gas is actually diffusing into tubular lumen. This ammonia will combine with the H plus, which is coming from this side, and the H plus will form the ammonium, and the Na plus, which is found there in the lumen, is actually diffusing back using again the symporter present here. Here there is a symporter, so they can exchange these things, and this ammonia which is there is trapped and it is excreted. So as you can see, ammonia is a very acidic thing. Yes, so the body is able to actually get rid of a lot of acids by this mechanism, and also parallelly, if you see, the sodium bicarbonate is the alkali is also recovered at the plasma site. So these are the two things happening. So if you to sum up the third strategy, bulk of the acid load is excreted in the form of ammonium ions. So that is how ammonium is very important to the body, because, and some scientists consider ammonium as a major urine acid. So some scientists say that this is a very prominent mechanism when compared to the other strategies. So so some call it ammonium as a major urine acid, and as you can see, yes, there is a net loss of ammonium, net loss of H plus again in this, and also net gain of bicarbonate is also happening at the plasma site. So you have all this now. Now moving on to the last strategy, which is, yes, this is the last strategy. This happens in a very negligible amount when compared to other three strategies. Again, the same reactions happens. There's no change. The first reaction would be this, then splitting of these into these two, and we have H plus crossing this side, sodium crossing that side, and we have bicarbonate being recovered there in the plasma, and H plus is excreted freely. So that is why the strategy is now named as excretion of free H plus. Now and also at the parallelly you are able to recover the alkali which is present there. So to sum up, it is very negligible amount is lost like this, right? So that is the only thing to remember. And also, as you can see, alkali is again recovered, and also H plus is getting excreted. So that is the fourth mechanism or fourth strategy. Now, if you speak about the location of various mechanisms, I told you this is these four strategies occur in the kidney in different parts of the kidney. As you already know, the nephron itself can be divided into the Bowman's capsule, proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct, right? So if you ask me where are each of these strategies occurring in different part of the nephron, these are the locations, right? So the excretion of free H plus and the first mechanism, both of these are occurring in the PCT, in the proximal convoluted tubule, whereas the titratable acid and the ammonium excretion occurs in the distal convoluted tubule, or some of them occur in the collecting duct also. Yes, so today in this class we covered the other two mechanisms or the lines of defenses of our body employed against acid-base imbalance. Respiratory regulation, which happens after some minutes of blood buffer action. Now with this also body won't stop, or if it is not corrected by this, body will actually change the strategy to this renal regulation. Under this we learned 4 strategies. One was the recovery of filtered bicarbonate. Second one was the recovery of filtered bicarbonate happens, then excretion of titratable acids, excretion of ammonium ions, and excretion of free H plus. Okay, these are the 4 strategies which comes under renal regulation. And yes, next class we will be talking about yes, this is not that. Next class we will be talking about acid-base disturbances, anion gap, and metabolic acidosis. So with that, we come to the end of second class. So the next class, that is third class in the 4th class session series, next class we will be discussing on these topics: acid-base disturbances, anion gap, and metabolic acidosis. So thank you.